Awesome. Can you hear me? Yeah.
Yep.
Perfect. So thank you so much for the invitation. I am always really excited to talk to you about the coral snake venoms, which are my passion. I've been working with this for about 12, 13 years now. So I'm really happy to come here and talk to you about them. You already said I work at the Institute of Biotechnology in Mexico at the laboratory of Dr. Alejandro Alegon. And here we work on venom characterization and mainly on the analysis of antivenoms and how they work against different snake venoms, but also some spider and scorpion venoms. So just a very, very brief introduction on coral snakes. They belong to the family Elapidae. They are one of the most diversified elapid groups in the world. So they have about 90 species of coral snakes in America. They are relatively small, 40 to almost 1.5 meters, but that's a huge coral snake. They're usually not that big. They are phaeophagus, semifossorial, and all of them are venomous, even though we have a lot of non-venomous colubrids that call false coral snakes and are extremely similar in terms of size and color. But coral snakes, the real ones, belong to the family Elapidae, and they are all venomous. Their venom apparatus consists, like all other snakes, of a venom gland, which we can see here, and compressor muscles, which, as their name suggests, compress the gland, and the venom is injected through grooved teeth, which are here, and they have a proteroglyph dentition, which means that the teeth are in the front of the mouth and they are fixed. They're like cobras, like mambas, like all other elapids. So in terms of the amount of venom that they can produce, it's relatively low when you compare them with vipers, which can inject much more venom. With experience on venom extraction, we have observed that they can go anywhere from not giving any venom at all, or less than 0.1 milligram, to over 35 and 40 milligrams per extraction. That's a lot for a coral snake, but just for comparison, medium-sized viper can give you over 400, 500 milligrams per venom. A big-sized viper can give you maybe 900 milligrams, something like that. But the venom is extremely toxic. So even though they don't produce a lot of venom, the envenomation can get pretty bad for some species. So envenomation is not very common in North America, or in America in general is not very common. In Mexico is extremely uncommon. About 2% of the 4,000 about 4,000 snake bites that occur in Mexico are from coral snakes. But still, when they happen, they can be severe. They are characterized by a neurotoxic clinical syndrome, which usually starts with the muscles of the face and causes flaccid paralysis in general. So muscles of the face will start relaxing and stop moving eventually. That can go in the face and then cause double vision, muscle weakness, salivation, in severe cases, flaccid paralysis of limbs, and it can be fatal. In really extreme cases, but still it can be fatal. So local pain and swelling can be present, though it is relatively mild, but in some cases it can cause a little bit severe pain. There is a very variable asymptomatic window, so the symptoms can start after maybe half an hour after the bite, or sometimes even 12 to 13 hours after the bite. So what happens is that sometimes people just think that they were bitten by something that was not venomous, and they don't go to the hospital because they don't really feel anything. And after a long time, then the symptoms starts happening. It is important to know this because people that get bit by the snakes should be in observation because it can get really bad if they are not treated. And currently, the only effective treatment is a specific antivenom. So what about the venoms? The venoms are composed, like all venoms of all other snakes, they're composed of a lot of different molecules, most of which are proteins and peptides. And all of the relevant toxins in coral snake venoms in mammalian venomation are acting on the neuromuscular junctions. So we have two main types of neurotoxins. The first type is alpha neurotoxins, which belong to the three-finger toxin family of proteins, and they block the neuromuscular, the nicotinic acetylcholine receptor. And this is what causes a flaccid paralysis. They prevent acetylcholine from binding, and therefore the muscle cannot contract. And we have the other type of neurotoxins, which are beta neurotoxins, which belong to the huge family of PLA2s. These beta neurotoxins, what they do is they inhibit the acetylcholine release, and they eventually cause a destruction of the presynaptic button. And this prevents the acetylcholine from getting released to the synapses, and therefore they also cause flaccid paralysis. So they have two mechanisms within the venom that will block neuromuscular junction and eventually cause flaccid paralysis. So this sounds relatively simple, but most of the coral snake venoms in America are not studied. We have only studied about 50% of the venoms in total. I say we in a very general way, but most of the studies have been performed in Central and South America, particularly in Brazil and Costa Rica. And it has been observed that most of the South American species are really abundant in three-finger toxins, while North American species and some Central American species tend to be more abundant in PLA2s. So this is a general trend that hasn't actually been confirmed, but it is very important in terms of antivenom neutralization. An antivenom that has been produced using a PLA2 abundant venom is not going to neutralize a three-finger toxin abundant venom in general terms. And as you can see, Mexico here we have a map that is showing only the species that have been studied in terms of their venoms. And as you can see, Mexico is almost empty. It doesn't mean that Mexico doesn't have species. Actually, it has around 15 different coral snake species, but until about 10 years ago, they had just been absolutely unstudied. So here I'm just going to tell you a little bit of what we have been doing in the last 10 years in terms of characterization of the venoms. So this is just a very cartoon map of the distribution of coral snakes within Mexico, and the colored ones are the ones that we have been studying in terms of their protein. So here we can see a few really interesting stuff. First of all, we have the venom of Microagus, and as you probably know, there are two genus of coral snakes in America, Microagus and Microagus. Microagus is the most is a sister clade for all other Microagus. So the study of their venom is really interesting because it can tell us something about the evolution of the rest of the coral snake venoms. And interestingly, and even though Microagus is distributed in North America, in northern Mexico and the southern United States, it has a venom that is very abundant in three-finger toxins. So this makes us think that the ancestral state of the venom was three-finger toxin abundant, and that the PLA2 abundant venom is a derived trait. This is just a very initial idea, but there's evidence there. And the rest of the venoms, or at least most of them, this is Microagus aticularis, this is from central Mexico, and it is extremely abundant in PLA2s, as well as the Microagus fulvius, that's the one from the United States, from Florida. And Microagus tenor is kind of a half and half type of venom. So here we have some initial, some general idea on the composition of coral snake venoms in Mexico. But one of the main, of the very important things here is that this composition does not imply that the main toxins on the venoms are PLA2 or three-finger toxins. So within each venom, we only have 2 or 3 toxins that actually cause mammalian venomation. So what we have been doing is separating venoms and trying to identify the main toxins that cause envenomation. And what we have been finding is that in each venom, we only have a little, well, a relatively small percentage of the venom that is actually responsible for mammalian venomation. And this is extremely important because these are the toxins that will be or that should be targeted by antivenom in order to create antivenoms that are actually working and that are more efficient than the ones that are currently available. So we have Microagus tenor, which I was telling you is something like half and half. So it's got three-finger toxins and PLA2s that are important, as well as Laticularis and Brownie. These 3 have both three-finger toxins and PLA2s that are relevant in envenomation. Microagus fulvius is interesting because it's almost exclusively PLA2. It doesn't really have any three-finger toxin that is relevant for envenomation, even though it does have three-finger toxins that are present. And Microagus is the other way around. So the toxicity of Microagus venom is almost exclusively caused by a single alpha neurotoxin that comprises about 18% of the venom. So these are the toxins that should be targeted by antivenom, and we've identified them in these species. We still have a lot to do, but this is the initial studies. And something else which is really, really important is these composition has been studied from 4 to 5 individuals. Each of these maps, of these graphs, comprises 4 to 5 individuals from very specific locations. So in terms of venom variation, we have very little information, not only from Mexican coral snakes, but other coral snakes. We have very, very little information in regarding the question, does the venom variate between one location and other? We know very little, but we recently have a very exciting opportunity to analyze ontogenic variation in these venoms. We had 11 Microagus diastema that were born at the herpetarium, and they were absolutely awesome. We had 11 eggs, and they all hatched, and we currently have them at the herpetarium. They're about 4 months old now. And we were able to extract venom from all of them. So we wanted to ask, at least to know if the venom variates between the neonates and the mother, to have a little bit of information in terms of venom variation in this species. And what we found is really interesting. We got a lot of venom from these snakes. These are 20 centimeters neonates, so they are really, really small. And we got from some of them, almost 0.5 milligram of venom. So that's a lot. And we found that there is very little variation between siblings. So this is an HPLC. You cannot see it very well, sorry, but this is an reverse phase HPLC, and you can see that there is little variation between the components, but the general pattern is very similar. That's the same here in the gel. But the mother, this is the mother, these are the individuals, and the mother is quite different. So what we don't know yet, and here is mother. Here is a comparison between the neonate, a pool of the neonate venom and the mother, and the venom is quite different between mother and neonate. So we don't know if the venom of the neonates is going to eventually change and become similar to the one of the mother, or if it's just going to stay like that. We are currently getting different venom extractions to see if that is the case. But so far we know that the neonate venom is about 2 times more lethal. I mean, the LD50 is about half that one of the mother. So that part is really interesting, and we are very excited on having this information. And well, that is what I wanted to tell you, and because I don't have a lot of time, but if you guys want to ask any questions, I just want to thank everyone that has been helping me. Obviously, this is not just my work, so thank you very much for listening to me today.
Awesome. Thank you so much. So you say you don't have a lot of time right now because there's quite a few questions, it seems, coming in. You want me just to ask the first 3, and I guess you can, or first 2 or 3?
Sure.
Okay.
No, I mean, I don't have time here. I could stay forever.
Oh, I see. Okay. Great. So the first question was, how are you testing the different neurotoxins in a mammalian model? Are you testing the venom in other systems other than mammals?
Yes, and that is really interesting. We have been testing them in vivo in mice, and we have also tested them when we are able to obtain a snake model. We test them in Phanopsis, which is a small earth snake. And at least with the Venom of Microwaves brownie, it is very interesting that the main alpha neurotoxin, which is similar to cobra toxin, is absolutely, it doesn't do anything to snakes. It's actually quite mammal specific, which is a very unexpected result, because we thought that the venom was going to be very reptile specific. But apparently it's not the case. Apparently snakes and mammals, if you test the whole venom, it's almost the same. It affects both mammals and snakes in a very similar way. But if you test some specific toxins, you only get a few mammal specific toxins, and you don't really get specificity. We tested, when we can, we test them in vivo. We did find one single reptile-specific toxin in microstenner, but it didn't appear to be very relevant in the overall venom toxicity. Yeah.
Very cool. So interesting. I don't work on snakes, so I'm always astounded. So how have you been able to find which type of alpha neurotoxins are in these species, i.e. short chain and long chain, and or do they have similarity to alpha neurotoxins from other species outside of the Americas?
Yeah, so we have been doing proteomic analysis. So we do have a primary sequence, and we have observed that they are very similar to short chain alpha neurotoxins from other elapids. And also there have been there are information on proteomics of Southern American microbes, and the most abundant ones are short chain alpha neurotoxins. We have also tested them on nicotinic acetylcholine receptors. So for those, we are quite sure that they have alpha neurotoxic activity. And yes, we have identified also other 3-finger toxins for their primary sequence. So we do have, they are very abundant in other 3-finger toxins, but they don't appear to be very relevant in mammal envenomation.
Interesting. So next, so for the PLA2 species, is this a single origin, or at least are all the PLA2 species each other's closest relatives?
Oh, I really wish I could answer that question. First of all, I'm not very definitely phylogenetics and phylogeography is not my expertise. So I wouldn't risk to give you a theory about that. What I know is that the most ancestral species appears to be three-finger toxin abundant. But the PLA2 abundant species are much more complicated because in North America, we have a PLA2 pattern and also in Central America, but those are apparently the most derived trait. So I wouldn't risk a theory on that.
My apologies. My cats were fighting quite literally on my lap. So
Oh, they do that.
Very interesting. So one other question from Sharon, and I might have a short follow-up to this as well. Given the variation in the different coral snake venoms within Mexico, how would you proceed with development of a polyvalent and anti-venom?
Oh, that is a very interesting question too. And I didn't have time to tell you a lot about it, but what we have right now is an antivenom that is very efficient to treat PLA2 abundant venoms. So what we have been trying to do is to include recombinant alpha neurotoxin antivenom. And actually that is the one that I skipped because I didn't have time to do it, but by including an already immunized horse that has been immunized with venom, with the complete venom, what you do is you try to include a recombinant alpha neurotoxin. And what we have found is that for many of these venoms, you get an improvement, a significant improvement in the case of tendon and diastema, which are the ones that are abundant in alpha neurotoxins. You get a very important improvement in terms of their neutralization. So what we need to do to get a polyvalent antivenom is try to enrich the low abundant proteins that are in the venom. So for PLA2 abundant venoms, you include recombinant alpha neurotoxin, and that way you make the neutralization spectrum much wider. So this is just a very small start for that, but in theory, you could just use a few PLA2s and a few three-finger toxins from Southern, Central, and Northern America. And if you select this correctly, you should be able to have a polyvalent antivenom. So we're going to be able to work. We're going to be working with that eventually.
Very cool. That's awesome. So mine is probably, I'm hoping it's a somewhat easy question. So it's about the ontogenetic variation. So I also work with cultured animals, and I kind of wonder all the time by giving them the same diet over and over if that's changing their venom. So I'm wondering, obviously not with this set, but perhaps in the future, if you got the chance to do this again, if you might change the diets of the younger ones to see if that has any effect, or do you all think it would?
It may. Well, with coral snakes, it is very complicated because there is not enough information on their natural history. So we actually don't know what they eat in the wild. So right now we are just feeding them forcefully with a I don't know the name of that, but by tube. So changing their diet in this case would be very, very complicated. We actually feed them what they eat. And if they eat, we're happy. So with other studies of snakes, of snake venom, there has never been observed that within the lifetime of a single individual, the diet would modify the venom. But there is a theory that the change between the diet in neonates and adults is what drives ontogenic venom variation. But I've never seen a study that actually concludes and proves that it's the case. It's they are always relation, but there are never really strong relationships. So apparently it's a very multifactorial change there. It's not like in some amphibians that is evident. With frogs, it's obvious that you change the diet and you change the poison, or I'm calling it poison this time.
Yeah.
But with snakes, there's never been this change.
Yeah.
It's much more complicated than it sounds.
Yeah, no, I can already believe that's very complicated. I don't work with vertebrates at all because they're complicated. Well, thank you very much. I think that's all the questions in the chat. So thank you. So our